Hi, I'm Assistant Professor John Chen. I'm based in the Department of Microbiology and Immunology, and welcome to my eOpen Day Talk. Today, I'm going to be talking about two things. First, I will share some of my own personal journey through academia. And second, I will tell you about some of the research interests of my lab. So hopefully some of you will find this useful, or at least interesting, and maybe it will help some of you make a decision about graduate school. So graduate school or not, I think most of you watching this video will probably be in your final year of university. And the only thing that's certain is that graduation is approaching fast. So I think some of you will probably join the workforce, or maybe you are thinking about it. And many of you are still trying to decide if graduate school is for you and if you want to do a PhD. Some of you have already decided that you want to do a PhD, but you don't know which one, which program. And some of you have already narrowed down your choices of programs, and maybe you don't know which lab you want to join. So what's clear is that there's a lot of choices. Do you want to do microbiology? You want to do immunology? You want to focus on basic science or translational, clinical? What country do you want to be based in? US, UK, Singapore, like here. There are many different options. But thinking back to when I was at this stage, the biggest question I had was, how does this whole process work? Meaning you go to grad school, you get a PhD, and then what? What do you do with that? What do you do after that? And to be honest, I really did not know. And when I got to graduate school, most of my classmates didn't know either. The only thing that we did know was that we were science majors and we like science. And so doing a PhD seemed like a logical choice, but we didn't really know where it would all lead. So there are many career paths that you can take, and we can spend a lot of time discussing them all. But today I will just tell you about my own abbreviated version and my own personal journey through academia. And hopefully for some of you, this will answer some of your questions, and it might help you decide if pursuing a PhD is right for you. So my story begins in university, and as a freshman, I had already decided that I would go to medical school. So I followed the pre-med path, and I majored in molecular biology. So the first year passed, second year passed, and by the third year, I was pretty well on track for medical school. I had taken all the required courses, I had done some internships, and at that stage, I was pretty certain that this is what I wanted to do. Then in my third year, I started my senior thesis project. And I started in the lab of Thomas Silhavy, who's a famous microbiologist, and he's also in the National Academy of Sciences. I spent well over a year in this lab, year and a half, and it turned out to be a transformative experience for me. So I learned a lot, obviously. I learned a lot of science. I learned how to do a lot of bench work, a lot of protocols. I identified several genetic loci involved in the regulation of the RNA that we were interested in. And then I wrote this up in a big thesis titled The Thermal Regulation of MCF RNA. But most importantly, I was introduced to independent research. I didn't particularly enjoy the biology courses in my major, but research was really something else. And it was something that resonated with me. So much so that it completely changed my career path. And at the last minute, I decided not to go to medical school. And I applied to PhD programs. So from there, I moved on to graduate school. And I moved on to Columbia University. And first, I joined the Department of Microbiology and Immunology. And I did 3 rotations in very different labs. In the end, I decided to join a host pathogen lab that was working on Legionella pneumophila. So Legionella is a gram-negative bacterial pathogen. And what's interesting about it is when it gets engulfed by phagocytic cells, it evades phagosome-lysosome fusion. And it does this with a secretion system called the type 4 secretion system that it uses to inject toxic proteins to disrupt the host cells. And so my project was to identify these effector proteins. And the effector proteins were the ones that allowed the bacteria to survive and replicate in the phagocytic cells. So in that lab, I spent many years, and I learned a great deal. Learned a great deal about gram-negative, the genetics of gram-negatives. I learned a lot of molecular genetics, protein purification, cell culture, microscopy. And in the end, we were the first to identify the Legionella effector proteins. And that resulted in a first author publication in Science. So when you finish graduate school, you can choose to enter industry or you can stay in academia. I decided to stay in academia, which meant doing a postdoctoral fellowship. So for my postdoc, I stayed in New York City and I went to the NYU School of Medicine and worked in a Staphylococcus aureus lab. That lab was led by Richard Novick, and there I studied bacteriophages, pathogenicity islands, all within the context of Staph aureus pathogenesis. So I spent many years there, and I learned all about the genetics of gram-positive bacteria, which, believe it or not, is very different from gram-negative bacteria. I also learned about the biology and genetics of phages, of pathogenicity islands, and there we did a lot of animal models of infection. So many of my projects didn't go so well, some did go well, and in the end, we identified a generic transduction of toxin genes, we identified a novel mechanism of gene transfer, and these different stories resulted in first author publications in Science and Molecular Cell. So just like when you finish graduate school, from a postdoc, you can once again choose to enter industry or stay in academia. And at this stage, I was pretty certain I would stay in academia, so I applied for principal investigator positions. And in 2015, I joined the faculty here in the Department of Microbiology and Immunology. So this last remaining part of my talk, I will give you a brief overview of what my lab does in my research program. So we work on Staph aureus, and why do we work on it? Well, because it's everywhere, and it's a problem all the time. It's everywhere in the community, it's everywhere in hospitals. It's a commensal and it's a pathogen. It lives on animals, lives on people, and it can colonize all different parts of your body. As for virulence, you name it, staph infects it, and it causes disease. So it can cause a wide range of diseases from necrotizing pneumonia to osteomyelitis to endocarditis. So my lab also is interested in bacteriophages, which are the viruses of bacteria, and they are the most abundant biological entities on the planet. They are important because they control the populations of bacteria, and they kill and they lyse them every day. And they also do something important in which they shuffle genetic material between bacterial cells. So they play a big role in the evolution of pathogens. And here's just a simple depiction of a phage life cycle. Here, all the way on the left, you see the number 1, the excision event, in which a phage genome has popped out of the chromosome and circularized. The same thing happens when the phage injects the genome into the cell. Then the DNA replicates, and then proteins are made by the phage. And eventually you have structural proteins, and it makes the capsid, and then the DNA gets packed into there, and the phage gets complete. But it doesn't get out till it lyses its way out. And when it does that, it kills the host, it lyses the host. So this is a very important step, and this is why phages control bacterial populations. So the research interests related to phages in my lab are that we are interested in engineering phages for safe use as antimicrobial therapeutics. This is also known as phage therapy. We're interested in novel aspects of phage biology and phage-phage interactions, and we're also interested in novel mechanisms of genetic transduction. And recently we published a novel mechanism of genetic transduction. And you can read about it here in this article, Genome Hypermobility by Lateral Transduction, and that was published in Science not that long ago. So my lab also studies pathogenicity islands. And in Staph aureus, we call these SaPls, for Staph aureus pathogenicity islands. And these are DNA elements that reside in the chromosome of Staph aureus. Most strains carry at least one. And they are important because they carry all sorts of terrible superantigens and enterotoxins. Plus, they do something, the particular family of pathogenicity islands, the SaPls, does something very interesting in that they are molecular parasites, and they parasitize bacteriophages. So they do something called molecular piracy. And what do we mean by molecular piracy? Well, you can see the same phage life cycle depicted in this cell on the top half, in which the phage is replicating and it makes protein and then it makes phage. Well, the SaPl follows a parallel pathway. It also excises out of the chromosome, starts replicating its DNA. But instead of making its own structural proteins, it basically just makes one or two, a handful, a very small number of proteins, and it steals all the rest from the phage. Okay, so this is essentially like hijacking the components, the structural components of the phage and stealing the capsid. And in a sense, it's not that different from, say, like stealing a vehicle, stealing a car, only it's a DNA element doing it, which we think is pretty cool, and that's why we study it. So for the SaPl research interest, we're interested in the mechanisms of this molecular piracy. We're interested in strategies by phages to counter molecular piracy, and we're also interested in mechanisms of SaPl-mediated gene transfer. So I hope this talk was interesting and will be of use to you in your decision-making process in choosing a PhD program. If you found some of my own lab's research interesting, feel free to email me, and we can talk about a rotation or something. Or if you're not interested in my research, in my department, the Department of Microbiology Immunology, there's many excellent labs doing very great work. Or on a wider scale, the School of Medicine has 18 departments and 2 centers to choose from. So thank you for listening, and good luck.